So, great. So you're seeing the presentation, great. And I apologize because when I get nervous, I tend to talk too fast. So I turned on the subtitles that will hopefully follow the presentation as well. And as you know, this talk is called Apples and Oranges, using comparative biochemistry to predict biosynthetic pathways. And these are my socials. I'm moving from Twitter to Mastodon. This is my Mastodon. And my group right now has three main areas of research. My group has, I think, 1 year of existing, and it has three main lines of research. Mass spec-based metabolomics. We have a QTOF that will hopefully be installed by January. Natural product biosynthesis with an emphasis in evolution of chemical diversity of the land plants, and data-driven biochemistry in what I call creative cheminformatics. And I'm sorry that I'm going to start with this slightly long introduction on data-driven methods, but I always like to introduce like the methods themselves to avoid any confusion. So hypothesis-driven methods is the classic way of doing science, right? It's the classic way of explaining the scientific method, which is you have an observation, you make a hypothesis in a way that can be tested, you do the experiments, you analyze the data, and if your data doesn't match, you change your hypothesis, right? And this is the way we do natural product chemistry in general. And this is the work that I work with Ornella Calderini and Sarah O'Connor, studying the biosynthesis of oleuropein, which affects the quality of olive oil. And one of the substrates is the oleurocyte methyl ester. And the first work that I'm going to present is quickly how we did it. So basically, we have the oleurocyte methyl ester. And the idea is, well, how is this biosynthesized in the olive? And so by analogy, we thought like, well, this looks quite like secoxyloganine. It's just the position of the double bond, what changes. So it may follow, by analogy to Catharanthus roseus, it probably follows the same biosynthetic pathway, or so we thought. And indeed, analyzing the transcriptome data, we found two enzymes that convert secologanine to secoxyloganine in the olive. And so I thought, great, now we just need something that makes the isomerization. And then we started testing for isomerases. And we tested tens and tens of enzymes with no results, no activity whatsoever. So again, the scientific method, we have the hypothesis, the data doesn't match, so we change the hypothesis. The problem is, which part of the hypothesis is wrong? Should I change from E. coli to Saccharomyces? Should I move to Benthamiana? Should I try a different kind of enzyme? And this, for example, and I am so thankful that this got published after I solved this, because otherwise I will be looking for oxyglutide-dependent enzymes, which actually catalyze a very similar reaction. What part of the hypothesis is wrong is one of the key problems to find out in natural product chemistry, in looking for enzymes in discovery. It turns out it was a different substrate. So the same enzymes with, when we fed 7-epiloganine, they oxidize to ketologanine, and then one of the enzymes, the secoxyloganine synthase, synthesizes secoxyloganine, and the oleurocyte methyl ester synthesizes the oleurocyte methyl ester. So no isomerase needed. It was just a different substrate. And of course, the oleurocyte methyl ester synthase is highly expressed in cultivars compared to wild-type olives because it's related to flavor, and it makes sense that we selected for flavor in olives, right? But the point is, the hypothesis is a very key part, and sometimes it's not that straightforward to figure out what part of the hypothesis is wrong. So formulating the hypothesis is key in gene discovery. And that's the problem with hypothesis-driven methods. And I'm sorry that quoting Hermann Hesse, is like when someone seeks, then it easily happens that his eyes only see what he seeks, and he's able to find nothing, to take in nothing. And I took this from a nice review from Janayan Lerker, that the title is very strong, that is, a hypothesis is a liability. Sometimes when we have a hypothesis, we just narrow everything down, and we tend to not look for things outside of the hypothesis. And that's where data-driven methods come in. Data-driven methods focus on the point between observation and hypothesis. Is take as many observations as you can and create a model that generates a hypothesis that is likely to be true, right? And this is the point of data-driven methods, generating unbiased hypotheses efficiently. Because we can make any kind of hypothesis. We can say like, oh, you know what? This enzyme doesn't work on Fridays. So maybe we should test it on Mondays. That's a hypothesis that is testable. And I think some of us have actually considered. Um, but it's not like an effective way of making a hypothesis, right? And data-driven methods are methods that are inductive by nature. And I always tell my students, you don't go into metabolomics looking for answers. And that's general for all the data-driven methods. You don't go into those looking for answers, you go looking for questions. You go looking for very specific questions. And again, with germanophones, to ask the right question is half the solution to a problem. So data-driven methods focus on that, on finding the right question. And of course, the other half requires pipetting. So you always need to complement them with hypothesis-driven methods. And this is not new. Of course, since we have a lot more data, then we have more data-driven methods. But this has been happening since the experimental philosophy times of Isaac Newton's. He famously said, hypothesis non fingo, which is I come here with no hypothesis in his Principia Mathematica. And this is the quote that I want you to keep in mind for the rest of the presentation. All models are wrong, but some models are useful. So now, after a very long introduction, a very quick introduction on the Lamiaceae. This is my work. I was working with monoterpenes in the Lamiaceae because they are good chemotaxonomic markers. You can see this is the mint family, the Lamiaceae, and you can basically divide it in two clades. The smelly clade, which has oregano, it has catmint, it has lavender, mint, and they have nice smells because they produce a lot of canonical monoterpenes. But in the non-smelly part, which is also the non-profitable part that I'm working with, the carbon flux goes to non-canonical monoterpenes, which are the iridoids, and these are water-soluble. So these are good chemotaxonomic markers to divide the family in two, basically, the Nepetoideae and the non-Nepetoideae. But even within the non-Nepetoideae, some scaffold modifications of iridoids are happening only in very particular families. So you can use them as chemotaxonomic markers. And there has been a lot of reports on chemical diversity of iridoids, which is mainly based on scaffold modification and decorations. But the point is, can I get more of all this data? There's already a collection that relates chemical structure and a species, but the point is, what more can I get out of this? And the logic behind this work is, if you can connect all of the molecules via reactions, then you can draw a pathway. So that's one. And second, if you know the phylogeny, you can clean up the selection in a way that is very similar to solving chess problems, but in this case, it's a tic-tac-toe problem. If you know all of these moves, all of these winning moves have a common, come from the same gameplay and pathway, then you can choose intermediate plays to explain the highest amount of winning solutions with the lowest amount of intermediate steps. So that's like a maximum parsimony approach. And this can be extended, of course, to molecules. If we know that certain molecules are specific to certain species and we know the relation between species, we can connect all of the molecules with all possible pathways, but then just keep the pathways that require the least amount of assumptions and the least amount of new emerging reactions, right? Again, maximum parsimony. So first I need to connect the molecules via reactions. And the problem is that chemometric fingerprints measure similarity, so they tend to connect molecules that are similar. But I need substrate product pairs, which is not connecting molecules that are highly similar, but connecting molecules that can become one another, that are connected by one biochemical reaction. And I saw two solutions: turn artificial intelligence to predict biochemical reactions, which is going to be, which was moved to future research, or find a different way of codifying iridoids, which is what I did. Most of the iridoid diversity just is based on oxidation steps. So I decided to assign each carbon a number based on the oxidation state. So 1 if it's an OH, 2 if it's a ketone, 3 if it's carboxylic acid, 4 if it's decarboxylated. And then I can get most of the diversity explained in the molecule, but the problem was double bonds. So I decided to codify the double bonds as imaginary numbers, just because it was easier. And now I have each molecule as a point in space, in 8-dimensional space, which is a complex, in complex dimension. With that, I can actually get the distances are then proportional to the number of reactions that I need to convert one molecule to the other. So if I need one reaction, this is going to give me a distance of 1. If I need two reactions, then it's going to give me a distance of 2. And of course, this gives us biochemical complexity in distance from a pathway. So of course, then if I plot these distances in a heat map, then I get some really nice chemotaxonomic grouping. You can see it by the streaks in the same color, which makes a lot of sense. But we could already do chemotaxonomy. The point was moving beyond that. I'm going to take another quick break bear with me with a second, of how pathways evolve in nature. So in natura, in nature, you have an original pathway, which then through evolution, selection, and speciation varies in different species, but they still share the common ancestor. So some of the reactions of the ancestral reactions may still be shared. And these specific pathways were sampled through history, through historical exploration. Basically, there's a lot of reports of chemical diversity of, you know what, species A has compounds 1, 2, 3, species B has compounds 5, 6, 7. And the idea was to make an algorithm that could connect all of these results and generate pathway hypotheses that best reflect the original pathways. And I'm going to focus on this. This is the pathway reconstruction algorithm. Basically, it's a very naive reconstruction algorithm with 3 steps. First, you take all of the reported compounds. Then you expand all of the compounds by 0, 1, or 2 reactions. And then you connect all of the compounds to the origin. Of course, you expand by 1 or 2 reactions, you're going to have expected intermediaries that don't make sense. So you just connect everything to the to primary metabolism or to the one original scaffold. And then you have a lot of pathway hypotheses. You can see here you have at least 3 pathway hypotheses. And this will, of course, have a lot of positives, and this is very messy. So this is just like 10% of the data, and then I gave up. And you can see that this is not useful. So it's completely useless. Well, not completely, but not as useful as I thought it would be. So then we need to clean up this selection, remove as many false positives as possible. And for that, we use phylogenetic pruning, in which we take each species. You can see here species A, species B, and species C. And you can see that the general pathway is still here conserved. In solid, you have reported molecules, and in these shaded dots, you have the predicted compounds. So you take all of the species, and then you connect each one of the species to the origin. Then you assign each one of these predicted compounds a weight, a predicted compound or a predicted enzyme. So it can be in edges or in nodes, a weight depending on how many species have that compound. For example, this compound is only present, this hypothetic compound will only be present in 2 species, but this one is going to be present in all 3. And then you go as well, marking all of the compounds. And you can do this with either a phylogenetic correlation, which assigns a high weight towards the tips, towards highly related species, or with Felsenstein weights that do the opposite. They assign a higher weight if it's reported in faraway species. And then finally, you take the paths to the origin, that has a higher number of weighted reports. So basically, you can see here in the solid line, this one requires the least amount of assumptions because it only needs 1 compound that has not been reported in all 3 species to generate a pathway. But of course, I always like to add a tolerance, so we can add a percentage and say, like, you know what, that pathway is the most likely pathway, but these pathways are also possible, just to avoid false negatives, which are very costly. So now we have a pathway reconstruction algorithm, and now the problem is we want to get as many true positives as possible without getting too many false positives, which are not that bad, or false negatives, which are very bad in this case. So how do we choose the best parameters? Because we have 5 parameters to choose. How do we train our algorithm? And the problem is that we don't have enough data. We need a lot of data. We need a lot of complete, unequivocally complete pathways, and there is no such thing. Because if a compound is not reported in a plant, it doesn't mean that it's not there. It just means that people haven't found it. Maybe because, and most likely because they were not looking for it. And there's not a lot of fossil record of molecules. And of course, that's a joke. So that's in red. It's impossible to get ancestral states of pathways. So the only way to get ancestral states of pathways is to make our own data. So decided to make, inspired by the black hole picture, where they generated a lot of artificial data. They had an expanded dataset. We decided, well, let's just make our own evolution. And so this is what happens in nature. So ideally, we could go back in time. And but the problem is that even if you go back in time, even if you rewind evolution, then the slightest change will lead to completely different things. And there's no ethics committee in the world that is going to approve that. So we decided to do it in silico. So they say like, well, if we have a pathway in a computer and we use a nature-like evolution model, then we can generate a speciation of pathways. And we can do random sampling, emulating historical scientific exploration, just sample these pathways randomly. And then we can use an algorithm to predict the pathways. But now, since we know the original pathways, we can have an estimation and an evaluation. And this then becomes a reverse engineering algorithm, which is easier to train. Right? So if we do everything in a computer, we can have a decent training of the algorithm. And this is key. If the nature-like evolution model works exactly like evolution, then this will be a really good estimation. If it doesn't, then it's not going to be a nice estimation. So that's the key and the problem of it. So this is our pathway evolution. Sorry, I have to remove this. Because yeah, it's not starting. Anyway, this is an animation of the process, but it doesn't matter. It's based on three evidence-based assumptions, which is a forward cumulative evolution, which is there's a random exploration of the chemical space, but enzymes are only recruited if they have a substrate. So you have random reactions happening in nature, appearing through evolution, but the enzyme is only fixed if it has a substrate. We also assume that there's an enzyme diversity threshold, just because enzyme diversity is costly, even because of the energy or the toxicity. And so there must be an upper limit on the number of enzymes that are in a particular system. And this has been observed in hybrids. When you create a hybrid, you suddenly have twice the amount of genes and you have twice the amount of diversity. But before the first, or at least at the first generation, then the expression diversity goes down to having approximately 1.2 at 20% as much diversity. So it's very close to an upper threshold. And finally, a molecular clock dependency, which means that there are no silver bullets, because silver bullets lead to a linear pathway. So there's no one compound that gives you an evolutionary advantage. It's the diversity that gives you the advantage. So this is very particular for iridoids. And this means that the furthest away a species is from the common ancestor, the more rounds of catalytic exploration happen. And please note that this is catalytic exploration, not catalytic innovation, because the enzymes are not necessarily fixed. And this has an implied assumption that there's a finite limited chemical space. And this is a publication by Andrew Donnell, which happened at the time I was writing, which got me jumping because, as you can see here, the assumption that there's some random exploration of chemical space appears to be happening in the case of caffeine. You have some enzymes, some enzymatic activities that appear randomly, and they are only fixed if they have a substrate. So this activity completely disappeared in the Paulinia, in the Spindales, because it didn't have a substrate. Although this activity got fixed in citrus, in Camellia. because it did have a substrate. Right, so this makes sense within the model, and this was completely independent and happened when I was writing, and I was jumping when I read this paper because you're like, oh, then it makes sense. And of course, now we can generate different evolution of different pathways, in which we also know the ancestral states. And I'm going to hurry up because I took too much time. So now we can generate pathways, and the problem is, how do we know which parameters recreate evolution better? Because again, this process needs to be very close to evolution for the evaluation to work. And yeah, we can create a lot of pathways depending on the pathway size, the ancestral pathway size, the maximum number of enzymes, the emergence of activity, and how much sampling we do. So we created a bunch of pathways and with different parameters, and now what? Like, how do we choose the parameters that best resemble nature? And it's similar to getting a multiverse of pathways. And well, maybe if we put something that we have an idea of what will look like in nature, then we can start discarding pathways, right? That are not like this pathway. But of course, we cannot compare pathways directly because we do not know the pathways. That's what we're trying to get. So we compare the conjectures themselves. The hypothesis is the thing we are comparing. And so we take the pathways, because we generated thousands of multiverses, so thousands of sets of 21 pathways evolved, the 21 iridoid pathways evolving. And so we selected the ones that had the most similar network parameters to the in nature reconstruction. So you can see here, this is an example of the ancestral reconstruction of the in natura pathway, of the real iridoid pathway. And you can see here one of the random points here. It's an in silico ancestral reconstruction of the pathway. And you can see that it's quite similar. Like the network structure, even if the structures are not the same, the network structure is very similar. But we have the great advantage that in the in silico pathway, we know which one of these enzymes, which one of these pathways is the actual real one. And we can say that, well, this reaction is not there. This is a false positive, something that we can never do in nature because we don't know if the enzyme activity is there, but just hasn't been reported. We do know that in the in silico pathways. So now, again, all models are wrong, but now we can know how wrong our model is, and we can train and choose the best parameters that give us the best positive predicted value, right? So then we run this in all of our iridoid data, in all of the real natural data, and we got finally an ancestral reconstruction of a pathway. We also have 21 pathways, one for each species, but we could also reconstruct what we predict to be the ancestral pathway. And long story short, again, we have 22 individual pathways comprising 87 metabolites, 23 of which have not been reported, and 122 pathways, 122 enzymes, and the prediction of an ancestral pathway. So that helped us to collapse all the available data. This was generated by the MINT consortium, MINT consortium, and we have 52 transcriptomes of all of the lamiaceae and 2 expression panels. So this is expression in different tissues in Lamium album and Calicarpa americana, which are the 2 species that are furthest apart in the tree. You can see here the distance between Calicarpa and Lamium album, so they are very far apart. But if this is the ancestral pathway, then at this point, the ancestral enzymes must have existed, even if the activity was lost. So based on this ancestral pathway and the assumption that the ancestral enzymes must have existed, we did the classic grouping guild by association approach, in which we just check the expression pattern of other enzymes in the biosynthetic pathway that are known to be in the biosynthetic pathway, and we selected all of the transcripts that had similar expression patterns, but we also just focused on the orthogroups that were in which the expression patterns were similar to the biosynthetic enzymes for Lamium album and Calicarpa americana, and that reduced all of the thousands of transcripts to 10 orthogroups, which also had a P450 activity because again, the diversity is based on oxidation reactions and oxidation of the scaffold. We tested with the predicted ancestral substrates, and we found an activity that was predicted by the model. Because again, you always have to pipette, you always have to pipette. So, and we found that this clade of P450s is enriched in species that are predicted to have this reaction. And again, we tested and the reaction actually happens. And the important thing is that neither the substrate nor the product are reported in Calicarpa. We found this reaction in Calicarpa because it was predicted in Calicarpa, but neither the substrate nor the product were reported in Calicarpa. So we find an enzyme completely de novo, and then we went back to Calicarpa to look for the substrate, and indeed it was there, just with a really tiny amount of it was being produced. But we found an enzyme that was an enzyme from a substrate and a product that were not reported in a species. And for me, that's kind of like the usefulness of the model. Because again, all models are wrong, but they must be useful for them, for it to exist, to be useful. And again, so this model converts reported molecules into a series of testable hypotheses. And more importantly, it tells us which metabolites to expect in each species. And for people doing metabolomics, that is like quite a statement, because the main problem of metabolomics is compound identification. So if you have a clue that a compound might be present there, then it's easier to find, as we did with the Calicarpa. No? And especially also, it helps to organize, contextualize, and rescue knowledge from the field. This is from reports of iridoid diversity from the 60s, and I don't think anyone reads any paper from the 60s anymore. So this already puts them in a format that people are familiar with, and it's summarizing a lot of information for avoiding for it to be lost. And then the current and future work, if funded, is to expand this to all the land plants, now training an artificial intelligence, and now doing a lot of automated, because all of these, the compound selection was manual, so we have to automate the collecting chemical diversity reports. And why for all plants? Just to spite funding agencies that say that this is too ambitious. But why not? If you're doing it for one species, why not for all land plants? And also trying to test close coevolution of enzymes. We can predict enzymes that are evolving closely. And this is a really nice case of coffee, because this enzyme must have evolved first in order to carry out the whole reaction because of Gibbs free energy. And that's basically it. This is the acknowledgement. This is the acknowledgements of all of the labs that I was working with, and involved are the people that directly participated in the project. Omar and Benji helped a lot with the collection and pruning of all of the structures. And yeah, Benke helped a lot with the synthesis of substrates. I don't think this could have been done without Benke and Jindy. Jindy helped a lot with the testing of enzymes. Robin Buell's group that handled the transcriptomes and sequenced Calicarpa and Lamium. And sorry, this is the group as I left, the Max Planck, and this is my group now. This is the two of us. And this is the analytical platform of the Centro de Tecnología, of the Centro de Biotecnología. And here you can see this box. This box has a QTOF that hopefully will be installed, and then I will have metabolomics capability here. And yeah, this is save the date. You're welcome. The next annual conference of the Phytochemical Society is going to be in the summer in Monterrey, in the Tec de Monterrey campus, which is a beautiful campus, to be honest. And yeah, and this is the paper. Please check it out. My idea was to get a discussion happening after, like this is a paper that will benefit a lot from post-publication peer review, from discussion of the scientific community. So I leave you here the name and the QR going to the paper. And I'm really happy to take any questions. Even destructive criticism is welcome. Like, yeah.
Fantastic. Thank you very much, Carlos. Questions?
I have a quick question. Oh, sorry. Should
Go ahead,
Yep.
Sam.
Okay. It's very beautiful talk.
Thank you.
You can extract hypotheses from the metabolomic data. I'm wondering, is there a way to match the enzymes that potentially catalyze these reactions from your pipeline to those to the hypothetical reactions?
That's the tricky part. This was done like the usual way of doing gene discovery now. So it's not very high throughput. This is low throughput. You go in and you know, like, oh, this enzyme is probably catalyzed by a P450. So let's look at P450s, and then let's look at if the expression patterns match, and then check if the orthogroups are shared. So yes, that is very manual, and that is very time intensive, and it requires a lot of effort. I still haven't figured out, and that'll be a nice addition to automate that. So I can actually give you instead of giving you hypotheses on pathways, I can give you hypotheses of enzymes. That'll be really, really useful, but we are not there yet. I still haven't figured out like how. We still depend a lot on homology for annotation of enzymes, but there's some new algorithms that are using structures for predicting enzyme activity rather than homology. So maybe one of those. But yeah, it'll be great if I could give you enzymes rather than pathways.
Thank you.
Gaurav.
Yeah, so a very nice talk. Thank you. My question was, I understood how you're generating the universe of reactions, but I couldn't get how you are narrowing down that universe into a small set of likely steps.
Basically, I am assigning weights to each. I am assigning weights to each reaction, and I am assigning weights to each predicted molecule based on in how many species do I need it. So in this case, this intermediary is needed in all of 3 species. So this will have a high weight. And this intermediary is only needed in 2 species. So this will have a lower weight because I will also need for this species, I will need to have a different intermediary. So that's the way that I am pruning. I have all of the possible reactions, and I am only keeping those that are needed in as many species as possible.
Okay.
Of course, it's not directly, because not all of the species have the same weight. Not all of the families are equally sampled. So I am also adding a weight either based, the one that worked better was phylogenetic correlation. So if it's needed in a closely related species, then you add a higher weight because it is more likely than 2 closely related species share the same intermediary. And that's the way I am discarding everything else.
Okay. Yeah, so I mean, the second part of that question was, since it's based on presence of the metabolomic data, and if you especially, I mean, mint family is very well sampled, but if you go across land plants, then there's a lot of sparse data generated using various instruments, various data generation methods, protocols, etc. And so how applicable will this algorithm be when you go larger evolutionary distances?
Yeah, and that's a very good question, because that's one of the biggest limitations. Even in the Lamiaceae, not all of the species are sampled the same. There are some species that are sampled a lot, and some species that aren't. So I try to not discard. Like, this is if something is predicted, then it's likely to be there. But if something is not predicted, that doesn't mean that it's not likely to be there. And that must be always the interpretation. That's a limitation of the algorithm that I cannot at least at this point, I don't know how to deal with. So I'm just considering that, yeah, especially in the land plants, it's gonna be there. The limitation is gonna be there. Hopefully, people will see like, oh, there's a really big vacuum in this family. So let's explore this family more because there's a vacuum. Like there's not a lot of data on this family, rather than this compound is not in this family. The right way of interpreting is there is no information of these compounds in this family. But yeah, that's a very real limitation of the algorithm. That some families are just sampled a lot, and some families are not.
Thank you.
Jörg.
Yes.
Great, thank you. Hey, Carlos, this was fantastic. I'm very impressed. I need to read
Thank
up
you.
on much more. So I don't know if my question actually makes sense or if it is perhaps not a good question. I understand that you're using these algorithms that you developed here for pathway reconstruction and pathway discovery, correct?
Yes, pathway reconstruction and gene discovery.
Right. So would it be possible at all to expand this in the future for development of new pathways that are new to nature? So if you can think about a metabolite that you would like to make in a bioproducts or bioengineering or synthetic biology approach, and you can use all this information that you're putting together here and the algorithms that you developed to reconstruct the pathway or discovery, right? To basically predict what enzymes would you need and which intermediates would you need to come to a desired new product.
Yeah, and I actually submitted funds. I think Professor Tuitang is here. We actually submitted funds for that. It didn't get approved, of course. But the idea was like, well, maybe if we get one of these intermediaries, maybe if we have a new to nature pathway that is in this space, maybe we can get something from the ajogoidy that is going to require less reactions to make a synthetic pathway, or that can give us an intermediary of that pathway, so we don't have to start from pyruvate. Rather, we start from this intermediary that we predict is going to be in this plant or in this family of plants. To make pathways by itself, directly, no. The part where we predict reactions, that will help. And that's what we were proposing for making new pathways. If we have reaction prediction algorithm, then we can just run this and minimize the number of reactions towards a natural point. So again, we just place the new compound in this space, and then we just follow the least amount of reactions to a point that exists in nature. That's the way it could be used for, or that we plan to use for synthetic biology.
Great, thank you. And thank you for getting me to go back to my bookshelf and
I saw the original quote.
and I found the quote actually. Great, thank you.
Yeah, so thank you. I didn't put it in German because my German is not that good. So my pronunciation is gonna butcher it. So
Lars.
Hi, thank you. Yeah, very interesting presentation. I was wondering, I have like maybe two questions. Like, first question is like, what kind of data sources do you need, and are you using to basically know which metabolites occur in which species? I mean, for the lamiaceae, it's basically kind of like sampled, but is that like kind of like primary literature, and you go through that and like basically record each metabolite manually, or is there like some way to do this more efficiently or on a larger scale?
yes, for the lamiaceae, this was done manually, and that's why I thank a lot Omar and Benjamin Lichtman helped a lot with that. They actually went to the primary literature and saw the NMR spectra and saw like, oh, well, okay, this one is likely to be true, and this one is likely to be an artifact of the extraction. And this was, yeah, with all the lamiaceae, it was done manually. There's a lot of manual work. For the future, you will need a lot of data mining. And I'm looking for collaborators on that. So if you're interested, it will require a lot of structure mining and data mining from papers, because of course it's going to be impossible to do it manually. And the databases that currently exist are very incomplete, like very incomplete for a lot of pathways. So even with the IRIDO, it's like, I think less than 5% of or 10% of the sample is actually in either of databases like Coconut or something like that. Because some reports are very old and they are not integrated into databases.
Yeah. Yeah, I'm thinking like, I mean, there's in most of the metabolomics databases, there's maybe like information about like the metabolite, but not necessarily like information, like meta information, like the species or the tissue or something like that. Yeah.
It's very hard to get. I'm trying to get from the Dictionary of Natural Products, is the one that has that kind of information. Coconut sometimes has species in which it was reported, but the dictionary is very incomplete, and Coconut doesn't have that lot of information. So it will require data mining, definitely.
And then the other question I had was more about enzyme promiscuity. If how does the algorithm like deal with that? If there's like maybe one enzyme for multiple reactions, so it's like a constraint, or is that like or is it like yeah, accounted for?
Yeah, that's not accounted for. I do it. It's the same. I'm still doing it at the reaction level rather than the enzyme level. So I cannot tell you which enzyme. This was the enzyme selection was done manually.
Okay.
Well, of course, with some self-organizing maps and all that, but it was done the way it's usually done. If I want to predict enzyme, then I will have to consider enzyme promiscuity, which could also be a nice niche. Like if you have a common ancestor and the enzyme has not varied much, then it's probably making the same reaction with two different substrates, and we can search for I don't know, for example, this reaction that is a hydroxylation of this carbon, and this reaction that is the hydroxylation of the same carbon, maybe it's done by the same enzyme, and we can start trying that. I haven't, but this model allows for that kind of hypothesis to be generated.
Okay, thank you.
You're welcome.
I think there's time for one more question, if anyone has one.
I have a question, but this may be off base. So just trying to understand the method. Okay, so you're trying to piece together like how many steps it is to get from one compound to another. And then I was thinking about the fact that like some enzymes can do two things, right? They can like add two hydroxyl groups or something. How does that affect the model, the ancestral pathway estimation? Does it matter at all? Maybe it doesn't.
So far, I'm not taking that into consideration. I'm just assuming that the react, because actually this, sorry, I'm going back. Oh, I don't have it here. But yeah, this carbon follows an oxidation directly from some enzymes oxidize all the way from the carbon to the carboxylic acid, and this is just one enzyme. I am considering it as 3 reactions. That's also one of the that's also one of the drawbacks of this method, is that I cannot tell how many enzymes are between the substrates. I can only tell how many reactions. But this oxidation is very commonly happening in nature with just one enzyme. This is the whole thing is just connected as one enzyme. And one enzyme catalyzes these 3 reactions, and a homologue in the ajugoide just catalyzes these 2 reactions and leaves the aldehyde. And it's in both cases, it's just one enzyme.
So do you think it's safe to say that probably it doesn't affect the ancestral reconstruction of the pathways, because those steps need to happen somehow, but it's more when you're going to look for the enzyme, you can't match up directly the number of steps to the number of enzymes, sort of.
Exactly. Yes. Yes, because the reconstruction is happening at the reaction level. I consider this to be one pathway and this one to be another pathway that actually happens. Sorry, I was moving the other way around. Where did I leave this pathway? Sorry, it's a lot of slides. There you go, here. You can see here that this one is this one is carboxylic acid, and this one is an aldehyde. And you can see that in the ajugoide, the oxidation is incomplete, and so it goes through a different pathway because of an incomplete oxidation. If I was considering this as a single enzyme, then it will be really hard to get this this branch, because this is probably the same enzyme, just that in the lamioide, the enzyme carries the full oxidation, and in the ajugoide, it doesn't carry the full oxidation. So it's probably one enzyme, but it's two reactions.
Well, I thought this was really neat. I also have to read the paper more closely. I'm really excited about it.
Thank you.
Well, thank you again, Carlos, for a fantastic talk. I think there's a lot of enthusiasm about this type of work, and it seems like it has a lot of potential if we can get the data together. All right, I think that's all we have for today. Please tune back in 2 weeks when we'll hear from a scientist from the Kew Botanical Gardens. Should be an exciting talk, and we'll send email reminders. If anyone has any announcements that they would like us to share prior to the following talk, please don't hesitate to email us. Have a good weekend, everyone.